Good evening, everyone. I'm a special virtual seminar series, Demystifying COVID-19, presented by Pacific Northwest National Laboratory. This session is being recorded, so those of us who are not able to join us today can view it at a later date. My name is Jim Sanford. I will again be your moderator tonight as we hear this exciting third presentation in our 5-part series designed to share what we've learned and answer questions related to the global coronavirus pandemic that has affected us all in various ways over the past year. Before we get started, we'll go ahead and have our usual poll to start us off so we can get an idea of where people are joining us from today. There's a link being provided in the Zoom chat. You could click that from your computer or mobile device. That will take you to our polling feature. And as you enter your answers, we'll be getting some live time results populating the screen. So we'll take a moment here and let everybody start typing in where they're tuning in from. And I would just like to remind you that please use the link that's in the chat feature of Zoom or shown now here on the screen. If you type where you're calling in from into the Zoom chat, it actually doesn't pop up on the screen. So it's kind of nice to get more words on that word cloud. Here we go. Well, it's cool. Looks like we have another really nice turnout today from all across the country. I'd be hard pressed to say I remember everywhere that's popped up in the previous 2 polls. So I think I see Indiana that I'm not sure jumped off the screen at me last time. So welcome. Thank you guys for participating in that. It's always nice to see, see what sort of spread we've got on this online forum. So we can jump back to the presentation here. Typically, this is where I'd take a minute or two to describe PNNL's role in advancing the DOE's primary missions and our economic impacts. However, in an effort to streamline a little bit more tonight and save time for questions and answers, I'll just remind everyone in the audience that more details on those subjects were presented in the previously recorded talks that are available online, and it's also available through PNNL's website. So if you're interested, please feel free to visit pnnl.gov. I would, however, like to again highlight the ways in which Patel and PNNL strive to impact the communities where we live and work, and in particular, this Community Science and Technology Seminar Series. A link to the PNNL events page is displayed on the screen and is being provided in the chat window. That's where you can find more information about upcoming talks, and also the recordings from the previous talks of this seminar series are posted there. Typically, this Community Science and Technology Seminar Series is presented by PNNL researchers focusing on the research they do here. But as we've mentioned, and I hope everyone is familiar with, this is a 5-part series where we're doing something a little bit different and trying to explain and focus on some various areas of COVID research. So we've recruited 5 PNNL researchers to give presentations on the indicated topics. And each talk, again, is very beneficial on its own, but the series does have a bit of a flow to it and builds upon each talk. So if you haven't seen the previous 2, I would encourage you to visit the web and watch those and hopefully join us for the next 2 talks. And as an added bonus, there's a survey at the end after each talk. And if you complete all 5 of those, you get a digital certificate of completion. So far, we've heard great talks from Stephen Wiley and Amy Sims. Again, I hope everyone was able to join us for those, and if not, the recordings are posted on the pnnl.gov/events website. Today, we're going to be hearing from Katrina Waters, who will be discussing what we know about the transmission and some health effects of COVID-19. I'll introduce Katrina in a little bit more detail in just a moment, but first let me just remind everyone of the format of the presentations of this seminar series. Katrina is going to give a 25 to 30-minute presentation, after which we'll have time for some question and answers. I encourage you all to type your questions into the chat window as you listen along. These will be collected and presented to the speaker at the end of the talk as time allows. We've had a really great number of questions put in the chat over the last 2 weeks. The only downside, in my opinion, is that we haven't had enough time to get to several of them. So to help remedy that tonight, Katrina has volunteered to stick around for an extra 15 minutes or so. So we're not going to end right at the 6 o'clock time. And hopefully that will allow us some extra time to help address some more of the questions coming in. As usual, we're joined by a lovely panel of community representatives to help spur the question and answer series, or question and answer session. So please allow me to introduce our panel again. We have with us Loanne Ayers, the President and CEO for United Way of Benton and Franklin Counties. Loanne, how are you tonight and what are you hoping to learn?
Hey, I'm enjoying the sunshine, but I'm really looking forward to learning more about transmission, both distance and duration. It's something we all wrestle with.
Perfect. Next, we have Kate McAteer, Vice Chancellor for Academic and Student Affairs at WSU Tri-Cities. Kate, how are you tonight?
I'm well, thank you, Jim. So I'm really looking forward to hearing more about possible mitigation efforts, similarly to what Loanne was talking about, but especially in school environments.
Excellent. Next, we have Martine Valadez, the Regional Director for Heritage University's Tri-Cities campus and the Interim Executive Director for the Tri-Cities Hispanic Chamber of Commerce. Martine, what are you looking forward to learning tonight?
Hello, everybody, and thank you for joining us. I'm really interested in what folks that can't stay home and have to work out there in restaurants and other places, what they can do to mitigate, the spread, even if they do wear masks, etc. So that's really what I'm interested in.
Very good. And finally, we have Justin Rafa, the Artistic Director of the Mid-Columbia Master Singers. Welcome, Justin. Welcome. A few remarks.
Good evening, Jim. As a choir director, my typical work environment includes large groups of people standing shoulder to shoulder at times. So I'd like to hear more about the science behind why social distancing works and why 6 feet became the magic number that we've now heard referenced for months.
Wonderful, thank you. Thank you guys all for your attendance today. I think Katrina has a great presentation that should address those topics that you mentioned. So without further ado, it gives me great pleasure to introduce Katrina Waters. Dr. Katrina Waters is the Division Director for the Biological Sciences Division at PNNL and leads the Computational Modeling Core for the University of Wisconsin Center for Predictive Modeling of Infectious Diseases. Her research interests focus on understanding exposure to toxins, the mechanisms behind bioactivity, and network modeling of host pathogen interactions. She received her PhD in biochemistry from the University of Wisconsin. Welcome, Katrina.
Great, thanks, Jim. And I want to thank everyone for joining us here tonight. I'm really looking forward to sharing with you some of the science behind transmission and mitigation approaches that can be used to protect yourself and others from spreading the COVID-19 virus. So starting off with just kind of an overview in thinking about how we have been approaching these discussions so far. We have kind of a viral infection life cycle shown on the screen, and today's discussion is really going to focus on transmission and infected states of the virus within the viral infection process as people are infected and expose other people to spread the virus through the community. So starting out with some terminology that is important for everyone to understand. When we refer to a pandemic, we're referring to an outbreak of a disease that is prevalent over a continent, many countries, or over the world. When we say an epidemic, we mean a disease that is more localized to a single country. Transmission is spreading the disease, whether it's spreading it from person to person or from its reservoir, which is where it resides and where it grows, to people. And when we say that the virus is infectious or contagious, we are talking about basically the state of that virus being able to transmit the disease from person to person or from the reservoir to another reservoir or another species. We've heard a lot about mutations that have happened within the virus and different strains that are being identified. I'm not going to talk a lot about that today, but you'll hear quite a bit about it at the next talk, so stay tuned for that, as well as more information about vaccines. So vaccines are preventative measures to help build immunity against a specific disease. And then when we refer to a model, what we're really talking about is a representation of a disease or a process that recapitulates many of the aspects of that virus or that disease. And typically speaking, we mean a computational model, or we could even mean a cartoon kind of a model that shows how that might work. So in focusing tonight on my talk on transmission, transmission really is a measure of contagion and how we scientifically refer to a virus as being contagious. You have probably heard in the news about the R value or the R naught value, for example, and this is a reproduction number that is scientifically used to characterize the transmission potential of a virus. So, for example, if an R value is 1, then that is the average number of people that one sick person will infect. If the value is 1 or less than 1, then a pandemic will pretty quickly die out because the number of new infections is less than the number of existing infections. As the R value increases to then we describe that virus as being more contained, will grow over time to create a larger epidemic or pandemic. So in this slide, what I'm representing is that if you think about other viruses, you would compare the SARS coronavirus 2, which is the causative agent of COVID-19. The R value is between 2 and 3, as it has been measured around the world. However, that number can be different based in different places, based on the strains. And as you can see in this chart, how it compares to other viruses, yes, it is higher than influenza that we would typically see, but it is actually much lower than many other viruses that have been seen around the world and for many, many decades, such as measles and mumps that have a very, very high R naught value, which is why they are highly contagious childhood diseases and why vaccines have been so important to curtail those diseases over many decades. So how does the SARS virus infect a cell? You've seen lots of pictures of the viral particles with these little spiky proteins all over the outside. That spike protein is the protein that interacts with proteins on the surface of the host cell. And when we say a host cell, we mean a human cell or even species that would get in. And there are primary proteins that interact with those spike proteins on the virus surface to help activate that protein and help facilitate the binding of the virus to the cell membrane, eventually taking that viral particle into the cell. One of the major noted here is this angiotensin converting enzyme 2 receptor, or ACE2 receptor, we call it. That is one that you've also probably heard a lot about as kind of this primary protein that helps mediate infection in host cells. So once the viral particle comes into the host cell, it releases its nucleic acid, which is RNA in the case of this virus, and it uses the host cell machinery to replicate the virus and create more virions, which are the viral particles, individual viral particles. And what's really interesting is that one cell can produce many hundreds or many thousands of viral particles, which then get released from the cell and have the potential to infect neighboring cells or other cells and other tissues within the body. So I know you've heard a lot about the acute effects of SARS viral infections. So I'm not going to go through a lot of that in terms of, the respiratory infections and the short-term effects. But what I am going to talk about are more of the long-term effects that are really starting to emerge as we get months and months into this disease. For example, long-lasting lung damage is being seen in some of these patients where they continue to require oxygen, for example, supplemental oxygen in order to breathe because they have fibroid changes within their lung that actually inhibit the ability of oxygen to move between the blood vessels and the lung cells. Heart disease and heart impacts, heart failure, and heart attacks have been seen in COVID patients. One reason why is because heart muscle and many of these tissues actually contain this ACE2 protein. You've probably heard of angiotensin blockers or ACE2 inhibitors that are used to control blood pressure. This is the same protein that the virus is using to be incorporated into the cell. And so because these proteins exist on many other tissues, including kidneys, skin cells, gut cells, and that's why many of these other tissue types being affected. So in the case of the heart, we're seeing a lot of heart damage. About 30% of surviving COVID patients have some level of acute kidney damage that is seen in the hospital. This is partly believed to be the result of some of the blood vessel impacts, but also can be caused by different changes in blood pressure, as well as direct inflammation to the kidney cells themselves. Skin damage is another thing that we've heard about. People have rashes, itchy red skin, even blisters, for example. Again, that's thought to be caused by immune response and inflammation, as well as changes to blood pressure. But there's also something known as COVID toes. If you haven't seen pictures of it, you can look it up online. And again, this is kind of a red blistered skin infection of the feet that are seen in some COVID patients. Neurological damage, again, is another target that we're starting to hear a lot about long-lasting effects that can happen in some people that have been infected with COVID-19. Fatigue, sort of mental tiredness, for example, is something that's noted by many people where they just kind of feel like they're not fully there or can't think straight. Some people have indicated hallucinations or even some mental instability that's been caused by the SARS infection. And even the loss of taste and smell is the result of the infection of the tissue in the olfactory area at the back of the nose that is up against the brain tissue. And then blood clots and stroke is another impact of COVID-19. We're seeing quite a few reports of people who are having strokes, either in the short-term duration after being infected or in longer term. For example, there are many reports of folks that have been infected with the virus. They've kind of recovered, and then a couple months later, maybe they have a relapse because there is this damage that has happened somewhere in the body, and that damage just doesn't resolve completely. So one of the very common questions that we get is, why are some people who are infected asymptomatic? And this is really what is one of the things that is very unique about this pandemic that we have. SARS coronavirus 2 affects many people in such a way as they don't even know that they're sick at all. And even those who do show symptoms eventually can be infectious when they are pre-symptomatic. So for example, the day before they exhibit symptoms, they are actually infectious and can transmit the virus to other people. So why might that be? There are many hypotheses for why that might be, and we don't know all of them for sure. One primary reason might be that they have low virus levels in their bloodstream or in their lung. This could be caused simply by the fact that they were wearing a mask and they were exposed to a lower amount of the virus, and so their body is able to fight it off before it gets to be at a high level. Could also be because you are predisposed to have less infection ability. So for example, there are some reports that have indicated blood type is associated with your ability to become infected, such that type O blood might be more resistant to infection than type A or B blood. Some people have a very strong immune system and their body is able to clear the virus more rapidly, and that keeps the viral numbers down, but it also kind of keeps the immune response in check, because there's this balance between the immune response to kill the virus and the inflammatory response that happens after that, which can have damaging effects to your organs as well. So there's this balance that is important to keep in mind. Another phenomenon that scientists are pretty aware of and physicians talk a lot about is disease tolerance. So there's quite a bit of literature to describe a genetic predisposition in some people to infections, where their body can simply tolerate an infection without becoming diseased from it. And there are many reasons why this might happen. Again, whether they have a very strong immune response or whether they have low levels of inflammation in their system, and so they can actually tolerate a certain amount of inflammation without becoming sick from that level of inflammation. There are reports of partial immunity in people. For example, some people who've never been infected with SARS-CoV-2 are showing some strong antibodies and immune response to coronaviruses that they may have been exposed to in the past that are not the SARS-CoV-2. Many cold viruses are actually coronavirus strains, even though they are much less contagious or deadly than the SARS coronavirus. But there are some reports that being exposed to different types of common cold viruses that are coronaviruses could cause some partial immunity. There are also some reports that find some correlation between childhood vaccinations, such as polio vaccines, and resistance to infection with SARS-CoV-2. And finally, underlying conditions, as we've seen with a lot of the reports of mortality resulting from COVID, really is one way that you can become more susceptible to the virus and to the effects of the virus. But it's actually possible that an underlying condition could make you more resistant. So for example, there are underlying conditions such as asthma and allergies that have a natural tendency to downregulate your ACE2 protein levels, and because of that, make you a little bit more resistant and make your cells a little bit more resistant to infection from the virus. And so again, one of the unique things we've learned about this virus, which is different from influenza, is that asthma does not appear to be a very strong risk of severe COVID infection, and it could be because some of these underlying conditions are downregulating in some people their levels of the ACE2 protein. So how is COVID-19 transmitted from person to person? The primary route of transmission is through airborne droplets. You've heard a lot about droplets and how, for example, respiratory droplets can get expelled, like when you sneeze or when you cough. And these airborne particles hang in the air and they fall and they stick to surfaces, for example, or they get on your hands because you put your hands in front of your face when you cough. And then the particles contain those infectious viruses that then can get transmitted to other people, whether you walk through it in the air and you breathe it in or you get it in your eyes, or whether you shake hands with somebody who has it on their hands, or if you touch a surface like a door handle, for example. We know that the SARS coronavirus is viable and can live on surfaces for several hours, depending on the temperature or the humidity or the amount of sunlight that may be there to kill the virus, so that you may be able to detect the nucleic acid there from the virus, but the virus may actually be dead and no longer infectious. So that's why, for example, We don't necessarily clean an area a week after somebody has been in a room, because after a week, we know that the virus is very, very likely already dead if it is a dry environment or a hot environment, or if it is outside. And so it is really these more kind of indoor areas where you have regulated temperature and humidity, where the virus is likely to live for several hours or even a couple of days. So why does social distancing work? So the transmission from a sick person to other people really depends on the size of the droplets that are being expelled from your mouth or your nose, because the larger particles are going to fall to the ground more quickly than the small particles, which can hang in the air for a longer period of time. As I mentioned, humidity is an important feature because these droplets are wet, and as they dry, the virus will die because it is a protein. And temperature, as the temperature increases, the virus is going to be killed by the heat. But one of the major influences is going to be the velocity for which that droplet is actually expelled into the air. So you can imagine if you sneeze, it's going to go much farther than if you cough. But it's also really important to think about, for example, just standing and breathing, you are going to expel particles as you exhale. If you are talking, you're going to expel more particles than if you're just breathing. And if you're singing or if you're laughing, you're likely to expel them farther than if you're just breathing. And so this is why some of the regulations you've been hearing about in the guidelines on distancing have kind of changed over the last several months as we've learned more about this virus and how it compares to other viruses. One of the important things to keep in mind is that when regulations and guidelines were released last March, they were based on what we knew about previous viruses like influenza and decades of research that had been done on influenza to understand its life cycle and how long it survived on surfaces and how far it would be transported into the air. And now over the past year, we've learned so much more about this virus that we're able to take that science and change the guidelines accordingly. So why 6 feet, right? Even the WHO has said in some cases 3 feet is sufficient. And again, last March, the science for the 6 feet was based on computer simulations and observed videos of people sneezing and people coughing and watching these droplets fly. So for example, as I mentioned, the large wet droplets are going to fall sooner. And as long as you're not standing right in front of somebody, those will fall and you won't necessarily breathe them in. But the very fine small particles are going to fly through the air much farther and much faster, particularly if you sneeze. And again, depending on the humidity of the air, they could linger in the air for a while. And so 6 feet at that time, last March, was believed to be far enough that you were of lower likelihood to breathe in a particle if you walked directly through the path of where somebody sneezed, for example. But is that really far enough? And, you know, the answer is sometimes it is far enough, and maybe sometimes it's not. Again, if somebody sneezes in a room and it creates a cloud of particles, if you walk through that cloud right after they've sneezed, then you could particles into your nose, your mouth, or your eye. The masking is so important because if you can that route of the transmission, you can risk of receiving the virus. But the distance is always going to help. And that's something that we talk a lot about within the scientific research community, is the time you're exposed to something, the distance you are from it, and any shielding that you have in front of you. Those are the three things that can help keep you safer in that circumstance. So why were restaurants closed down? And why are restaurants really any different from any other indoor environment? Well, the reason why is because even during the previous SARS outbreak, there were documented cases of transmissions in restaurants. And this has been true for the current pandemic as well. So early last year, before the coronavirus was really well understood in terms of what it was and where it was and how to detect it, there were documented transmission cases in China of people sitting in a restaurant. For example, in the picture displayed here, the initial carrier in this example, and this is based on a publication, was asymptomatic on that day, and they developed symptoms later that evening. So they went to a restaurant with their family members, didn't think anything of it. But within a few days, the people sitting at their table became infected, as well as people at adjacent tables. And when they further investigated this, what they discovered was that there was an air conditioning unit at one end of the room, and the flow of air through that room actually matched the pattern of the transmission that they saw from the infected person to the adjacent tables. And so, again, thinking about air conditioning units, fans, and airflow within a room is going to have a really big impact on the ability of the viral particles to be transmitted from person to person. And so, again, very early in this outbreak, the restaurants were shut down because until we understood more, we wanted to prevent that from becoming kind of a hotbed of transmission within the community. So one way that our restaurants have been able to start reopening, particularly locally in the Tri-Cities area, is they were allowed to do outside seating, and particularly then indoor seating, where they had a window or a door open at two ends of a room so that air could flow. And one reason for doing that is obviously that if you can keep the air moving more rapidly, then you can replace the air with fresh air on a regular basis and keep purifying the air in the environment. What I'm showing you here in this slide is a really great article from the New York Times from a few weeks ago, where they reviewed the data from a paper where folks take computational models, and Tim's going to talk to you a lot about this in the 5th seminar series. And what they do is they create a mathematical, but also a spatial model. So a 3-dimensional model of a classroom or a building, and they can model the airflow within that building in a 3-dimensional way, and also with the impacts of temperature, the impacts of humidity, for example, or the impacts of simply having a room heater or a fan within that room for how the air will flow. So for example, in this figure, what I'm showing you is in a classroom, simulated classroom, an infected student at one end of the room where all the windows are closed, and they model, even though these students are separated by 6 feet, if you look at the transmission and the flow of air around the room, you can see that nearly all of the occupants of the room are going to be exposed to some levels of particulate viral particle across the room space. However, if you, in the second panel, have one window open, for example, it may concentrate all of that viral particle kind of plume, we would call it, around that one student and minimize the exposure of other students as it would circulate around that open window. And then in the third example, the final example, what they're showing there is the impact of having an air purifier at the other corner of the room, so that the viral plume from that one student can basically be blown up and over and out to that purifier and really minimize the concentration of that virus overall in the room itself. So masks, right? Masks have become a big thing. And the reason why simply is because they are a physical barrier that reduces transmission from person to person. A lot of people say, I don't believe in masks, you know, or I'm not worried about myself. So if we think about transmission risks and rates when we are wearing masks, the cartoon shown on this slide gives you a really good representation and way to think about this. If two people are standing together talking and one person is infected, there's probably about 100% chance that that person's going to get infected because this is a contagious and highly infectious virus. If the person who is not sick wears a mask, they do reduce their transmission by about 30%, but the plume of particles are still swirling around their head, and it's still likely that those particles are going to get on their skin, maybe in their eyes, maybe on their hands, and then they're going to touch their face later, right? So it doesn't completely block or entirely block or protect yourself to wear a mask. What is really important is that infected people wear the masks because that is the physical barrier that blocks the droplets from getting in the air in the first place. Now, depending on how well your mask fits, on how accurately you wear it, people who wear it below their nose basically eliminate the effectiveness of the mask altogether. But it is important for everyone to wear a mask because people who are asymptomatic could be carrying it. And if we all wear masks, we reduce the risk of transmission and the rates of transmission really, really dramatically. And we've seen that in the Tri-Cities area where the mask mandates came out and the rates really dropped. And then maybe people got a little bit complacent and stopped wearing their masks and the rates went back up again. And then as we get more vigilant, you know, the rates come back down. And so masks really do appear to be a primary way of protecting yourself, but more so protecting other people. So why do masks work? Well, as I said, it is a physical barrier, right? And mask materials work in two different ways. It can be simply a mechanical filter where depending on the density of the fabric, you get more or less particles going through it. Or some materials, like you may have noticed, like wool will give you a static electric kind of response. That electrostatic magnetism of a material can actually cling and bind particles. And so when scientists have evaluated the effectiveness of these masks, it really depends on both of these factors, but it also depends on the size of the particle. So, for example, very large particles are going to be blocked by most materials, but very small particles are not going to be blocked by materials that are breathable, because if they are breathable, that means air is going through and the particles are coming with it. So, for example, in the table that I have shown here, based on data from a scientific publication, if you look at 80 thread per inch cotton, which may typically be used in like t-shirts, for example, one layer of that cotton is really only 14% effective at blocking the small particle sizes that we see from a sneeze, for example. If you look at a denser cotton, like a quilting kind of cotton that is 600 threads per inch, you get up to 98% with just one layer. Many people would argue that's not very breathable, and that's true, but the more breathable your fabric is, the less effective it's going to be. Again, fabrics like flannel have big gaps in the threads, and so it's a little bit more breathable, but it may have a little bit more electrostatic kind of impact, so that if you start adding layers, like 2 layers of flannel, you get an enhanced amount of filtration and blockage of the viral particles. So what we're showing here in this table is the fact that sometimes combining different fabrics can give you a more maximal kind of coverage if you combine high filtration with high electrostatic, for example, in a multi-layer mask. Many people love their gaiters because they're breathable. But if you take a single layer gaiter and you look at the effectiveness is only about 50%, if you get a 2-layer gaiter, most of the time those are no more effective because the manufacturer has actually used more breathable material to make that 2-layer gaiter as breathable as the 1-layer gaiter. And so if you really wanted to increase the effectiveness in using gaiters, use 2, because then what you're doing is you're using a breathable gaiter and another layer of that same material to increase the amount of blockage and filtration that you see, as opposed to just wearing a 2-layer gaiter, which is probably more breathable material and just less effective anyway. So finally, I think, many of us are extremely hopeful about the impact of vaccines and how that is going to slow the spread within our communities. You've heard a lot about the term herd immunity, and I'm sure Kristen will talk a lot about it at the seminar next week. But if you just think about vaccines as umbrellas, and the more people who hold up a vaccine, the less people are going to get wet from the rain. If we can get 70 or 80% of people to get the vaccine, then we can really dampen down the transmission that we can have in our community, and really try to get this pandemic under control. And I think, we're now starting to see that in our healthcare workers that have been vaccinated. The rates amongst healthcare workers are coming down pretty dramatically. And the rates in the elderly are also coming down as the vaccine is getting distributed to those communities and to the retirement homes. So what has the community been asked to do? And we've been asked to stay vigilant through this thing. Wash your hands, and wash your hands, and wash your hands. It's always been a primary route of transmission, even of influenza. It's remarkable, our influenza rates this flu season are way, way down. And why? Because people are wearing masks, people aren't leaving home when they're sick, and people are washing their hands. It's great. Social distancing is still so important because, that is where we're going to transmit to our best friends, to our family. We still need to socially distance and keep our contact circle small so that you can reduce the number of people that are in your circle that could potentially transmit the virus to you unknowingly. Please get vaccinated. Childhood illnesses have dropped so dramatically in the world after we've had all the vaccines that are distributed. We've seen resurgences of measles in our communities, in our schools, because people are not getting those vaccines. Please get vaccinated because that will help everyone. And really stay vigilant. We really need to see this through. This is not going to be the last pandemic, and it's going to take a while as we see the different mutant strains coming through, and as people get reinfected. And we are seeing people get reinfected over and over again. And again, that is kind of a new thing that we're seeing with this coronavirus that makes it maybe a little bit different than other kind of typical viruses, or just other types of viruses that have been seen. So with that, I'm just going to close and say that, our next Tuesday is going to focus on different routes, why you get different answers, and the different vaccines, how they're different and how they work. And Kristen Omberg is going to be presenting that talk next Thursday. And then the following week is going to be Dr. Tim Scheibe to talk to you about computational models and how we use them. And with that, please submit your questions to the discussion chat, and I'll be happy to hang on and answer if I can.
Perfect. Thank you, Katrina. Thank you for doing my job there and promoting the next two talks. We'll mention that again at the end as a reminder, but really appreciate it. And again, as Katrina just mentioned, feel free to continue typing questions into the chat. We have a really nice turnout tonight, just shy of 200 people. I think it's got about 190 joining us, and there's been quite a few questions coming through. So let's jump right into that Q&A session. Let's start with our community representatives panel. So Loanne Ayers, if you're there and would like to ask your first question.
Thank you. Among the nonprofits that are serving the senior community, they're seeing a lot of seniors who have been vaccinated just kind of ignoring all social distancing. So the question is, if I'm vaccinated, why would I still need to wear a mask and practice social distancing?
Yeah, that's a really great question. So the answer is that the vaccine may not prevent you becoming infected. The vaccine may prevent you from becoming very sick from the infection. So, for example, even as we've seen with flu vaccines, over, many years. Sometimes they pick the right strains and sometimes they don't pick the right strains. But even when they don't pick the right strains, if you get the flu vaccine, you usually get less sick than you would have otherwise. And so, with this virus, what we're trying to tell people is that you could still become infected and then infect other people, but that vaccine will help keep you healthier because you should be less diseased from the infection, and it should kind of dampen it to a certain degree.
Great, thanks, Luann. Let's move to Kate.
Thanks, Katrina. That was a great presentation. So I don't know if you saw last week, but the New York Times published an article that 3 feet of distancing appears to be acceptable for schools. And so I was wondering if you read that and could you comment on the science behind it?
Yeah, absolutely. So I did, and I thank you for bringing that to my attention because I hadn't seen it this weekend. Yeah, so it's a really interesting study, and what they've done is they've looked retrospectively at almost 300 school districts and whether they had policies for 6 feet of distancing or whether maybe they couldn't enforce a full 6 feet. So they went with 3 feet because that was what they could manage. And what they saw was that the transmission rates among students and among staff were roughly about the same between the 3 feet and the 6 feet of distancing as they recorded from that data. What's important to keep in mind from that study is that, for one thing, there were very few school districts that were, I would say the fraction was only about 20% of the school districts that were included in that study had the 3 feet of distancing. So it was kind of an unbalanced statistical study, so it may not be fully representative. The other thing is that that study included both elementary schools as well as middle schools and high schools. Our elementary schools were able to open sooner because kids can be in a cohort within an elementary environment where they don't go room to room and they're exposed to only those 12 people for a whole day. And the cohorting helps you keep more kids safer. When you get to the middle school and the high school environment where they all go room to room and they go past each other in the hallway, I would argue that 3 feet or 6 feet for the desks probably matters less than the fact that they're all touching each other in the hallway, right? And so when you start thinking about it from that aspect, and as we've seen, because more schools have been open as elementary schools, the transmission rates in the schools are actually matching the community transmission rates, much more so than other places. But I do believe if we had middle schools and high schools open earlier and we had all of those kids mixing in the hallway, I have 3 teenage boys, they're not going to wear their masks the right way all day long, and they're not going to keep from touching people. So, there's only socially so much you can do to get kids to understand that.
Thank you, Katrina. We'll next take a question from Martine.
Hello, Katrina. Thank you again for your great presentation. So within the Latino community and others, there's many individuals who are essential workers and don't have the choice, like some of us, to work at home. And so they're exposed to others, and particularly perhaps to droplets from sneezes and others. So I want to know a little bit more about the droplets themselves, how contagious they are, and also how the length of transmission, how long do they have to, really watch out for that, et cetera. Thank you.
Yeah, so that really, the front line of the folks in the grocery stores and all of our essential workers and healthcare workers have really been the heroes in this pandemic because they put themselves in harm's way every day. What it really comes down to is their personal protective equipment that they can have with them, whether, they have just their mask or whether maybe they wear a double layer mask, for example. If they wear it really well and it fits well across their face, it's going to prevent, them from taking that in. Wearing glasses, for example, whether it's, safety glasses or sunglasses, can help prevent it from getting in your eyes. And then another big thing, like I said, is washing your hands and not touching your face. In the early days of the pandemic, when I would go to the grocery store, I had hand sanitizer in my car, and I would sanitize my hands before I left my car, and I would sanitize my hands the moment I got back in my car. And so I think just, this sort of constant kind of cleaning is really going to be the most effective barrier that people can use. But there's almost no way to prevent all, risk of transmission, especially if other people around you are not taking the same precautions.
Thank you.
Oh, sorry. Thank you, Martine. And Justin, we'll move to Justin to round out our community panel's questions.
Dr. Waters, this Monday, March 22nd, all of Washington State will advance to phase 3 of the Healthy Washington Roadmap to Recovery Plan, allowing for increased capacity for a variety of industries, including up to 50% occupancy for indoor dining. And although it's not required in the state plan, would it be advantageous for restaurants and other food beverage establishments to keep doors and windows open, as well as provide air purifiers to help limit any potential viral transmission?
Absolutely. I think without question, it would be advantageous if we kept windows and doors open, we keep air flowing, overhead fans that can blow air and continue it circulating. Many buildings, including office buildings, are doing additional external air turnover with their ventilation systems, trying to constantly bring in clean air to replace the indoor air. All of those things can make those environments safer. And particularly within a restaurant where people remove their masks and they're eating and they're drinking and they're talking, those are environments where, those particles can expose other people.
Thank you.
All right. Thank you to our community representatives for those questions. Katrina, I'm going to bounce a few questions off you that have been coming in from the audience. So there's been several related to Loanne's question, but I just wanted to touch on that again regarding if you've already been vaccinated, there's still the need to wear a mask, and a little bit, if you could elaborate a little bit more, what might help decision makers know when we're getting to a point where general mask wearing is not required anymore?
So probably based on community transmission rates, if we are seeing those rates go down dramatically until there are really only a handful that are happening. And if we can, at that point, trace them to how they're happening. Right now when we have, many hundreds of cases per week, the health department can't do a really effective job of tracing every single one of those for every single individual and all of their potential contacts to know where the transmissions are happening. But if we can get the numbers down low enough, and then the health department can identify where the hot spots are, that could certainly help us get to a point where we could more comfortably say there's less of the virus in our community to be exposed to. The other thing we can do is more testing. And this is something we've heard about for months and months. In the early days of the pandemic, we didn't have testing available at all to a certain degree. And then even when we did, we didn't have enough testing. And again, because of that, we didn't know where the virus was, and we didn't know how to keep people safe because it was kind of lingering. And so now that we have tests more available, now that we have more rapid tests, and if we can develop tests that can be deployed in a very rapid environment with very quick turnaround, then we could identify where it is and maybe more effectively allow people to start gathering. I know I've heard that they are making recommendations now for people who are fully vaccinated for them to get together within groups, because then all people within a group who are vaccinated are less likely to be carrying as high amounts of the virus to transmit to other people. Great,
thanks. Okay. So in quite a few questions related to surface transmission of coronavirus and COVID-19. I think that was one of the earliest things people did. As you mentioned, hand sanitizer was sort of one of the first things we started doing a lot. So in your opinion, or what the data is showing, how prevalent is surface transmission versus airborne transmission?
So we don't really know I would say, there are many scientific reports that are saying we have no documented cases of surface-based transmission. That is largely because we can't rule out the possibility of airborne transmission. If you have a sick person in the room and they leave the room and then other people come in behind them and get sick, there could still be airborne particles in the air for a certain amount of time. And if people are migrating from room to room, it's hard to eliminate the possibility that they didn't get close enough for an airborne transmission. And so while there aren't a lot of documented cases of surface-based transmission, there are many, many cases of COVID for which contact tracing was not able to identify a person-to-person route of transmission through an airborne particle. I will also say that in the previous SARS outbreak, there was a documented transmission from toilet fans in an apartment building. And we know that this SARS coronavirus is present in feces, for example, too. And so fecal particles can also be another reservoir for this virus and another potential for transmission.
Cool. Okay. Kind of related to that. So first off, is there a difference between airborne and aerosolized? Those terms, are they interchangeable?
To a certain degree, they're kind of interchangeable. We typically will say aerosols are the very, very small droplets that are going to go farther and hang in the air more than the larger droplets themselves, which are more likely just to settle more rapidly. So an aerosol really is indicating something that is much smaller and it's going to linger in the air longer.
Okay. And is there any idea exactly how long those aerosols may be able to linger?
So that's a really great question. It could be minutes, it could be hours. And it's going to depend on factors like the temperature in the room and the amount of air that's being circulated. So if you have a high humidity environment, it's going to be different than a low humidity environment. So for many of these particles, when we say a particle, we mean many viruses, little viruses. They're not in water. They're actually kind of in salt water because that's the fluid that's going to come out of your nose or your mouth. And many of those fluids, like even saliva, have proteins in them. And those proteins or cells that come from your mouth are protective of that virus. And so as we look at stability studies, if those stability studies are done in water, it's actually less reflective of the respiratory droplet, and the fluid makeup that is can actually protect the virus for a little bit longer. So in most cases, you're not going to see an aerosol hanging in the air for several hours, as they were talking about early in the pandemic, where somebody just walking down the aisle in a grocery store would breathe in something that someone sneezed an hour ago. That's probably much less likely to happen. And you're not going to breathe in enough of the virus necessarily to get sick. But on the other hand, depending on how susceptible you are and your own immune system, you could have a different amount of virus that is required to make you sick. And so, I hate to kind of be a little wishy-washy, but it really does depend on where you are, the conditions of the room, and who you are in terms of your own susceptibility.
Great. Okay, that makes sense. So a lot of people, as we saw in the poll, are calling in, listening in from around the Tri-Cities region. So we're a pretty semi-arid, dry desert environment. You've mentioned humidity a couple of times. In general, is humidity tend to make the aerosols last longer and be infectious for longer or less long?
So some of the data suggests that a high humid environment, typically because they tend to be higher temperature environment, will kill the virus much faster than a lower temperature, lower humidity environment. That being said, because we have a very arid environment, which is higher temperature and low humidity, the virus is probably less likely to exist outside for a long period of time at all due to a high temperature, a low humidity, and high ultraviolet exposure from the sunlight.
Great. Okay, thanks. I did just want to remind for anybody who is tuning in a little bit late, we are going to stick around here for about another 15 minutes. Katrina's graciously volunteered some more time to address some more of the questions that have been coming in. So we're not ending right now at 6. We've got a little bit longer to hang along. You talked about some of the longer lasting effects of COVID-19. I was curious if, you know, how long is long term? Are these going to be lifelong complications that people might suffer from? I know it's been about a year that this has been circulating. So are there people that have had complications for that entire time?
There are. I think people with underlying conditions for which they already have some disease or some damage to the tissues that can be affected by the virus are seeing long-term complications from this virus that are lasting months even. There are reports of people even who have lost their sense of taste and smell who never really got it back completely, or got it back maybe partly, but it's just not the same, or food tastes the same, different now to them, because there could be damage to their olfactory nerves that's impacting long-term taste. So yeah, there are. I think we don't really know yet the long-term impacts to our healthcare system, because we are discovering now that there were probably more infections earlier in January of last year than we recognized, because we weren't aware of how widespread the virus might be at that time, and we didn't have tests to know for sure. So there are people who have lingering effects from illnesses early last year who may not have been diagnosed at the time. They may have been tested for COVID later. The virus was long gone, but the damage was already done. And so now the medical community is having to kind of grapple with trying to understand on a molecular level and on a tissue level where this damage came from, so that we can then figure out how to fix it, or at least how to treat it to improve people's quality of life.
Wonderful, thank you. I think it might be a nice time to jump back into the community representatives that we have. I know that they're also staying around for this extra time, so give them a chance to ask a second question. Let's work our way backwards from our first one. So Justin, if you're there and would like to ask another question, that'd be great.
Dr. Waters, for the first few months of the pandemic, communal singing activities were deemed to have a high level of risk of transmission and were not permitted for some time. In January, the Washington State Performing Arts Sector Reopening Plan was approved that allowed for small groups of singers to rehearse together if they're all masked and stand a minimum of 9 feet apart, obviously a bigger distance than the standard 6 feet that we've discussed. However, this specific protocol around spacing of singers has recently been re-examined and is likely to be eliminated. My question is, if all people in a communal singing setting are masked, do you feel it would be safe for them to stand 6 feet apart rather than 9 feet, especially if those people are assembled in an outdoor venue? And I use the word safe in quotations, knowing that there's a wide range there of what we're talking about.
Yeah, so that's a great question. I think it could be, and I think, you know, if you think about it from the perspective, if they're facing the same direction, right? So that all the aerosols are going out there and they're not facing each other, right? It's going to be safer to be standing together. If their masks are effective, meaning that they fit, although I can imagine that being difficult when singing, because you have to be able to move, and moving your whole face necessarily, you would need a well-fitted mask so you don't have gaps on the sides, you know, by your mouth. But if you can find a good mask that gives you that room to be able to sing, but really holds that in, then I think certainly in an outdoor environment, particularly where you've got that kind of constant airflow, and even, you know, dry air and sunlight, I could see that as being, much more safe than being, in a closed environment, people facing each other and closer together.
Thank you very much.
Thank you, Justin. Martine, is there another question you'd like to ask?
So yeah, with essential workers and workers who are out there, they're wearing masks, let's say, and I saw you had a great slide there on the different types of masks and their effectiveness. My question is, so do the masks lose effectiveness over time, and sort of how do you know if a mask is no longer good to wear, basically?
Right, that's a really great question. Most fabrics are made to be washed many, many times. And so I think if a mask that's made from a good fabric, it's important to pre-wash the fabric so that if you have different types of fabric and they shrink at different rates, you don't wash it and then it's all kind of discombobulated and doesn't fit anymore. But certainly washing them is going to be a really great thing to do anyway. If you have the same mask and you're wearing it for weeks and weeks and months and you're not washing it, it could become less effective simply because of the moisture coming from your face, kind of clogging up that fabric. It'll make it less breathable, but may also make it less effective. Certainly, if you wash a fabric so many times that it's starting to break down, if it's becoming more breathable, it's probably less effective.
Thank you.
Thank you, Martine. Kate, you're up.
So Katrina, in higher education, many of the students are saying they really miss the social environment of the classroom. So, many colleges are looking at keeping large lectures via Zoom, but allowing students to come back in cohorts for, discussion-based sections or lab sections. So when Dr. Wiley presented in week 1, he had me thinking about that because he talked about movie theaters and similar to what you said to Justin, with everyone facing, the same direction. So it has me questioning this approach in higher ed to bring students back for discussion sections. And so I'm just curious if you have any thoughts about that, and maybe we should be flipping it.
Yeah, so that's a really great question. And again, I think, depending on the classroom environment, if it is a large lecture hall and they're distanced and if they're wearing masks, there is no more risk to them probably in that environment than walking around the grocery store and bumping into other people, right? I think, we lose sight sometimes of very specific examples versus many of the things we're already doing. The grocery stores have not been asked to open windows and doors and to ventilate because people have to eat and we have to have access to food. But we're getting exposed in those environments more so probably than we would get exposed in a workplace environment that's being done safely or in a classroom where everybody is masked, right? So I think from an educational perspective, you have no control over the activities of those students as soon as they walk out of the classroom. That barrier is probably going to break down at that point anyway. But I think for the case of the school environment, if you keep them in cohorts, and this is why elementary schools were able to open very effectively and very early, because you have the same kids, and if they have assigned desks, and if you know who they're exposed to, then you've got 4 or 5 kids who are going to be exposed and can be sent home if one kid gets sick, as opposed to 35 kids or even 75 kids who they may have touched throughout a day wandering through the hallways. So the cohort kind of model, I think, is pretty effective. And I've seen that be pretty effective so far with some of our school sports practices and stuff where they're trying to keep the kids in little pods, groups of 4 or 5.
Thank you.
Thank you, Kate. And we'll move back to Loanne. Loanne, do you have a second question for Katrina?
Unmute. Wow, that was fast. Indeed. We have people who are now eager to travel either for work or to see family members. And they may be highly likely going to places that have different protective standards in our community, like no masks in Texas or, different standards if they're going down to Mexico. What kinds of things could they do to better protect they and their children?
Yeah, that's also a really good question. So I would say, and as I've done in a couple of instances where I've been forced to travel, I bring along a better mask. I have my typical mask that I wear to the grocery store. But when I'm walking through an airport or when I'm sitting in an airplane, I do have a KN mask that I wear. It's got much better filtration and it's going to protect me much more effectively. I also stay away from people and I wash my hands all the time. So, you know, I take the corner seat in the restaurant, trying to be away from everybody with my back against the wall, and just try to protect myself as much as possible, for sure.
Thank you.
Great, thanks. Okay, I'm gonna try to rapid fire a few more questions to you to get some of the been coming in through the chat. Have there been any studies on whether or not there's adverse effects to wearing masks? I know that's information that's been around.
So, you know, I can speak from personal experience that if you're running up and down the stairway and you're wearing a mask, it can get hard to breathe. It can get really hot. And for those of us who aren't accustomed to wearing masks on a regular basis, it's kind of a hard adjustment within our daily lives. However, we should keep in mind our medical personnel, surgeons, you know, many doctors and nurses wear masks 12 hours a day. And it's about training. You know, they train themselves on how to breathe inside a mask so that they don't negatively impact their ability to breathe effectively. Certainly, you know, the surgeon who works on you for many, many hours is working effectively behind that mask. And so I don't think there are a lot of studies that have shown medical impacts of wearing masks. I think it's really uncomfortable for many people. For those who have claustrophobia, for example, it can induce panic attacks, I'm sure. And that is, of course, an adverse effect for those people that, you know, really makes it hard for them to wear a mask or to wear very non-breathable masks, for sure. But other than that kind of situation of it being extremely uncomfortable, I haven't read anything about adverse health outcomes from wearing masks. I would have expected to see that from the medical community before now.
Great, thank you. How about the R naught values that you were talking about earlier in your presentation? Is that calculated for like the United States as a whole? Can it be different region by region? How often are those numbers updated and recalculated?
Yeah, absolutely. Those numbers change all the time. So we've seen many, many reports of the R naught values for this virus. Very early on in the pandemic, there was speculation about the R naught value. So it is different from place to place, region to region, country to country. It can be influenced by the strain itself that is circulating within the community, but it can also be influenced by the mitigation approaches that are employed. So, for example, areas of the world that did not shut down very rapidly had higher R naught values than those who did shut down and implemented the isolation orders very quickly. Again, it is the average number of people that one person will infect. And so if you close schools, if you close restaurants, if you wear masks, you decrease that R naught value within a community.
Great, thank you. Any information on what the rate of reinfection might be if someone has had COVID-19 and, you know, whether or not they get vaccinated after having it?
That's a great question. So there are many documented cases of people getting reinfected over and over with the virus. Many of the cases that are documented in the medical literature are based on people with a compromised immune response. So the virus is maybe not coming to them independently from 3 different sources, but the virus is kind of hiding out in their system and then reemerging again later. And so we see these dormant periods of the virus as kind of what we call a latent infection, which then comes back again within an individual. However, there are also documented cases of people getting infected a second time or a third time. Again, that could be a new strain of the virus where your immunity that you got the first time was not complete enough to fight off the new infection or the new strain. Or it could just be the susceptibility. Maybe someone doesn't have a very strong immune response and the antibodies don't last that long. And that is something that was seen in the previous SARS outbreak, that antibodies to the SARS coronavirus only lived within people for several months and not for several years. And so it is possible that the antibodies will deplete over time and that the immunity could be lost. And so even for folks who have had the coronavirus, we advise that you get the vaccine anyway. There are reports that perhaps you only need 1 dose of the 2-dose vaccines instead of the 2 doses if you've been infected before. It may just be about how strong your body responds and generates those antibodies.
Great, thank you. Okay, last question for you. How about plastic face shields as a replacement for masks? What's behind that? Is that as good, as adequate?
No, really. So a plastic face shield is going to protect your eyes more than wearing a mask will protect your eyes. But a plastic face shield does nothing to prevent you from transmitting the virus to other people. And so if you remember the slide that I presented where the healthy person was wearing the mask, but the sick person was not, the mask only reduced the transmission by 30%. And with a face shield, it would be even less because the opening underneath that face shield is going to allow you to breathe in particles from underneath and they will get right into your mouth. So Great. You will see healthcare workers that wear a face shield and a mask, and that will increase your protection, but the face shield alone won't.
Great, thank you for clearing that up. Okay, we've hit 6:17, believe it or not. There's still a great number of questions. I really thank all of the audience engagement. Really thank you, Katrina, for sticking around for this extra time to help address some more of those. Thank you to our community representatives, and thank you again, everybody that's tuning in. Would just like to say a couple more reminders. There are 2 more talks in this seminar series. So next week, we'll be hearing from Kristen Omberg. She'll be talking about testing and vaccination. So there was quite a few questions related to vaccines that I hope will be addressed next week. I would also like to remind everybody one last time to take the survey after this. There'll be a link popping up in the chat window right now. So you could take that and that would be very useful. And yeah, 2 more talks to go. We're 3 down. Thank you again. It's been nice talking to you tonight, and I look forward to seeing everybody next Tuesday at 5 p.m.